Very good morning to dear students of class 9. We are dealing with the chapter number 6, seeds, structure and germination. Now in the last class we have mainly talked about the seeds, we have discussed about monocot seeds, the dicot seeds, we have discussed the structure of the bean seeds and the maize grain. Now today we will talk about germination. Clear? So before going into germination, all of you have seen the gram seed, am I right? Now, all the seeds, they contain a dormant embryo. Alright, so all of the seeds, before germinations, all of the seeds, they contain a dormant embryo. The embryo, it mainly consists of a radicle and the plumule. Radicle is the future root and the plumule is the future shoot of a plant that we have discussed. Clear? So all the seeds, they have a dormant period, or we can say the inactive embryo or the inactive stage. During this particular stage, the seeds will not germinate. Clear? So it's also called dormancy. Right? It's also called dormancy. D-O-R-M-A-N-C-Y. Dormancy, a period of rest where the seeds will not germinate. Now, when we see the seed externally, that means when we are observing the seed from its external appearance, we will observe that the seeds are inactive, they are without life. There will be no sign of any life when we see the seeds externally, clear, from the outside. But internally, a lot of chemical reactions or chemical activities are going on which we cannot see. Clear? Internally, those chemical reactions are going on. Now you can say that the dry seeds are consuming oxygen and giving out carbon dioxide in a very little amount, in a very minute amount. They are utilizing the reserve food materials in a very minute amount also, like those reserve food materials which are stored in the form of endosperm, or where the cotyledons used to store the food material. So these food particles will also be utilized in a very little amount, clear, when they are in stage of dormancy. Now, when these dormant seeds are placed in a suitable conditions or in a favorable conditions, then it becomes active. Clear? So when these dormant seeds, when they are placed in favorable conditions or in a suitable conditions, then they will become active. Now they start growing into a seedling. So whenever the seeds which are in dormancy, and when they becomes active, when they are placed into the favorable climatic conditions, and whenever they start growing to form a seedling, then actually we call it germination. Clear? So today we'll discuss about the definition of germination, we will see the conditions for germination, and we'll see the experiments to prove those necessary conditions which are essential for germination and the types of germination. Okay, so what is germination? So let's discuss it first. Now, germination is a process in which a seed starts growing to develop into a new plant. So you can see over here, so this is the particular seed. Clear? So when the seeds are placed in a favorable conditions or in a suitable conditions, then you can see it starts germinating. You can see the appearance of root, right, and the shoot portion which called the plumule part, radicle and the plumule, and slowly it starts germinating. So these the seeds which has been germinated into a new plant. So the process where a seed starts growing into a new plant is called germination. So in this process, a seedling emerges out from the seeds. So we can also define that germination is the process of formation of a seedling developed from the embryo. So this particular structure is the embryo, which is composed of radicle and the plumule. So from the radicle there will be formation of root, and from the plumule part there will be formation of the future shoots. So whenever the seeds start germinating, right? It will develop into a seedling, and the seedling will develop into a new plant. Clear? So remember the definition of germination. Now, what are the conditions necessary for germination? Clear? So there are lots of conditions which are essential for the germination of a seed. So from your textbook, kindly go through the page number 49. Clear? Page number 49, go through it. So mainly the conditions which are essential or necessary for germinations are water, suitable temperature, and air or oxygen. So these are very essential for germination. So we will go through it one by one. First of all, we'll discuss about water. Now, the seeds will absorb the water from the environment, or let's say from the soil. Clear? Because we used to pour the water in the soil, and from the soil, the seeds will absorb the water. Clear? Now, in a seed, in the last class we have discussed that there is a small portion just above the hilum. If you remember micropyle, small opening is there. Through this micropyle, the seed will absorb the water. Clear? Now, what are the main uses of water? Or what is the importance of water? So, the two main uses of water or the importance of water are: number one, the seed swells up, it will absorb the water. So when the seed will absorb the water, it will swell up. Clear? And at the same time, the seed coat will rupture. The seed coat will break. That means testa and tegmen, the two outermost protective layer of the seed. If you remember, we have done testa and the tegmen. These are the layers which provide protection to the seed. So they are called the seed coat. So the micropyle will help in the absorption of water, and this particular water which is absorbed by the seeds, alright, will lead to the swelling of the seeds and rupturing of the seed coat. The seed coat will rupture, and whenever the seed coat will rupture, radical comes out, alright, and it forms the root system. So radicals will come out, and that radical will lead to the formation of the future root system. Number one, use of water. Now number two, now water is also essential for the different types of chemical reaction that occurs within the seed, and at the same time for the activation of the enzymes. Clear? So those water which has been absorbed by the seeds, they are utilized by the seeds to carry out different chemical reactions and for the activation of enzymes, so that the reserve food materials in the seed, which are in the form of endosperm or which are stored in the cotyledons, can be turned into a diffusible form or the dissolved form, and those dissolved form of the food particles will be utilized by the growing embryo. Clear? So this is the significance of the water for germination. First use is that the water will be absorbed by the help of micropyle, and those seeds will absorb the water, they will swell up, and it will lead to the rupture of seed coat, testa and tegmen. At the same time, the radicals will elongate and will lead to the formation of future root system. And number two, those water will be utilized to carry out different chemical reactions and for the activation of enzymes within the seed. Clear? So that the reserve food materials which are stored in the form of endosperm or which are there in the cotyledons can be transformed into the dissolved form and can be utilized by the developing embryo. So this is the importance of water for germination. Now number two, suitable temperature. Okay, so let's check out the importance of temperature for germination. So mainly very low temperature and very high temperature are not suitable for germination. You can see over in point number one. Alright, very low temperature and very high temperatures are not suitable for the process of germination. We can see the reasons why. Because very low temperature inhibit means stop. Alright, very low temperature means inhibits means stop. So very low temperature inhibits the growth of the embryo, and very high temperature will destroy the delicate tissues. So very low temperature and very high temperatures are not suitable for the process of germination. So what is the suitable temperature for germination? So therefore, a temperature between 25 to 35 degrees Celsius. We can say moderately warm temperature. All right, between 25 to 30 degrees Celsius. Clear? Is suitable for germination. So this particular range of temperature between 25 to 35 degrees Celsius, between 25 to 35 degrees Celsius is known as optimum temperature. Clear? So optimum temperature means 25 to 35 degrees Celsius temperature, which is favorable for germination. So very low and very high temperature will not favor germination, but the optimum temperature between 25 to 35 degrees Celsius will favor germination. So it is called optimum temperature. Clear? So seeds of tropical plants often need a higher temperature for germination than those of the temperate regions. So kindly remember the optimum temperature that favors germination. Clear? The next one, oxygen or air, because air is also important for germination. So during the process of germination, there is a rapid cell division. You know what is cell division? Where the cell will divide and there will be rapid growth of the cell. Clear? So during germination, there will be rapid cell division and there will be a rapid cell growth. And mainly for this process of cell division and the cell growth, it requires energy. Clear? And energy is mainly available only by respiration. And for respiration, what is needed? Oxygen is needed. So therefore, oxygen is very, very important for the process of germination, because during germination, there will be rapid cell division, there will be rapid cell growth for which energy is required, and energy is only available by the process of respiration, where there will be breakdown of food particles, oxidation of food, and therefore respiration occurs only in the presence of oxygen. So therefore, water, suitable temperature, and oxygen are very, very important for the process of germination. So now we'll check out the few experiments on germination. So from your textbook, kindly go with the page number 50. Okay, we'll check out a few experiments on germination. So we have got in the page number 50 in the textbook, so kindly go through it. All right, and you may note this one also, because I have just subdivided the few things over here. So I have divided into requirements, procedures, observations, and conclusion or inference, because each and every experiments can be divided into a few subtopics, like what are the things you require to carry out the experiment, that comes under requirements. Alright, procedure means process. So what are the process that needs to be carried out to do the particular experiment? Clear? Then observation, what will we observe? Then after observations, we have to go with the conclusion or inference or result. Alright, so we will go to the first experiment on germination. So we will try to prove that water is necessary for germination. So I have told you that, go to the page number 50. We have got this topic, some experiments on germination. Clear? So first thing, so what are the things that we require to prove that water is essential or necessary for germination? First one, requirements. We require two beaker. You can see over here, beaker A and beaker B. Then we need gram sheets. Okay, green gram sheets, or we can say any other sheets you can take. Then cotton wool. One must be dry and must be wet cotton wool. Clear? So one dry cotton wool and the wet cotton wool. Now procedure. So take two beaker. So we are taking two beaker. You can see over here, we are taking two beaker, and we'll mark them as beaker A and beaker B. Okay, so we are taking two beaker and we are giving them beaker A and beaker B. Now in beaker A, what are we doing is that we are placing gram sheets on a wet cotton wool. Okay, so over here I'll do the correction over here. So this will be our beaker number B, and this will be our beaker number A. Okay, so in the beaker number A, what we are doing, we are taking the wet cotton wool that contains moisture water, then we are putting the gram sheets. You can see over here, these are the gram sheets. Clear? Then similarly in beaker B, now in beaker B, what we are doing, in beaker B also we are putting the gram seeds. Clear? But over here we are using the dry cotton wool. Clear? Instead of using the wet cotton wool, we are using the dry cotton wool in case of beaker B. So in what we are doing now, listen carefully. We are taking two beaker. We are marking them as beaker A and beaker B. In beaker A, we are putting the wet cotton wool that contains moisture water, and we are putting the gram seeds. Similarly, in beaker B, we are putting the gram seeds, but over here we are putting the dry cotton wool. Now both of the beaker should be kept in an ordinary room. Clear? Now you can see over here that beaker A contains moisture water, but beaker B there is no water, which is totally we are using dry cotton wool. Now what will we observe? So after a few days we can say that the seeds in the beaker A will germinate, but not in the beaker B. That means the seeds which is present in the beaker A will be germinated. Alright, that means the seeds in the beaker A will germinate, but the seeds in the beaker B will not germinate. Why? Now what will be your observation, will be your conclusion or inference? Your inference will be water is necessary for germination. Clear? Because without water the seeds cannot germinate. Clear? So it is very easy experiment. So what were the requirements? Two beaker, gram seeds, cotton wool, dry and wet procedure. We are taking two beaker, we are labeling them as beaker A and beaker B. In beaker A we have put gram seeds along with wet cotton wool, and in beaker B we have got the gram seeds, but we have put them in dry cotton wool. Then we have kept both the beaker in an ordinary room. After a few days we'll observe that the seeds in the beaker A will germinate, but the seeds in the beaker B will not germinate. What will be your conclusion inference? That water is necessary for germination. Without water there will be no germination. Clear? So you can copy this one. Clear? It will be easier for you. Number 2, experiment. Experiment to prove that a suitable temperature is necessary for germination. Now we are going to prove the importance of temperature for germination. So here also same thing. Requirements. Require two beaker, green gram seeds, wet cotton wool, and refrigerator. Clear? So here what we require two beaker, then gram seeds, then cotton wool. It must be wet. So there is no need of dry things over here. Then we require refrigerator. Clear? You can see over here refrigerator. So process. So same thing. We will take two beaker. We will mark them beaker A and beaker B. Clear? Now in both the beaker we will put the gram seeds. You can see over here we are putting the gram seeds. Then we are putting the wet cotton wool. So over here we are just using the wet cotton wool. Clear? We are not using the dry and the wet, but in both the beaker we are using the wet cotton wool. Clear? Now beaker A, this particular beaker, will be kept in an ordinary room. Are you getting it? And beaker B will be kept in a refrigerator. So beaker A will be kept in an ordinary room temperature, but beaker B we are putting this particular beaker into the refrigerator. You know the temperature in this particular refrigerator will be very less compared to the room temperature. Now observation. So after a few days, in a day or two, alright, the seeds in the beaker A will germinate. So those seeds which are present in the beaker A after a few days will germinate, but those seeds which were in the beaker B and which were kept in the refrigerator will not show the sign of germination. Though it will germinate, but it will take a longer time to germinate than the seed present in the beaker A. Now what will be your will be your conclusion or inference? That suitable temperature is essential for the process of germination. Clear? It's very easy. So this one also you can copy it. Clear? Now next one, we'll see the experiment to prove that air or oxygen is essential for germination. So let's try to prove that air or oxygen is essential for germination. Okay, so what can be done? Let's see now. This is very important. Requirements. Two conical flasks. You can see over here. first conical flask, second conical flask, then cotton wool, which must be wet. Okay, so you can see over here also there is a wet cotton wool. Soaked gram seeds, that means gram seeds which have mainly been put in the water for the overnight, we can say. That is very important. Okay, alkaline pyrogallic acid is very important in the experiment, because we have to know the importance of alkaline pyrogallic acid being used in this experiment. So what is the importance of alkaline pyrogallic acid? We discuss under the procedure. And we require the test tube. You can see over here, this one is the test tube. So these are the test tube. Okay, then we require a thread to keep the test tube in position. So let's talk with the procedure or the process. So we are taking two conical flasks. So we are giving their name as A and B. So we are taking two conical flasks and giving them name A and B. Now in both the conical flasks, we have to place the wet cotton wool and we have to place the soaked gram seeds. So in both the conical flasks, we have to put the wet cotton wool and we have to put the gram seeds. Now lower a small test tube containing alkaline pyrogallic acids in flask B. So we'll just now focus on the flask B. Now in flask B, what we are doing, we are putting a small test tube. Clear? By the help of a thread, we are keeping a small test tube in conical flask B, but that test tube contains alkaline pyrogallic acid. Now what is the importance of pyrogallic acid? It will absorb the oxygen present inside the conical flask. That means this alkaline pyrogallic acid will absorb the oxygen which are present inside the conical flask. So these conical flasks will be devoid of oxygen because alkaline pyrogallic acid will absorb all the oxygen present inside the conical flasks. Similarly, in test tube A also, we will put the test tube, in the conical flask A, alright, we'll put the test tube also, but this test tube, it contains plain water. Clear? So instead of alkaline pyrogallic acids, the test tube in the conical flask will contain the plain water. So keep the test tube hanging by fixing a cork on the mouth of the flask. So this one is the cork. You can see over here the black color in both the conical flask. These are the cork. Okay, these are the C-O-R-K, cork. Okay, so these are the cork. Now, what will be our observations when we keep these two flasks in an ordinary room temperature? So procedure one more time. We are taking two conical flasks A and B. Clear? In both the flasks, we have taken wet cotton wool along with the soaked gram seeds. In conical flask B, alright, in conical flask B, we are taking a test tube. That test tube contains alkaline pyrogallic acids. Alkaline pyrogallic acid will absorb the oxygen present inside the conical flask. So conical flask B will be devoid of oxygen. Similarly, in conical flask A, we'll place the test tube, but this test tube will contain plain water instead of alkaline pyrogallic acids. So oxygen is present in the conical flask A and is absent in B because it has been absorbed by alkaline pyrogallic acids. Now both the flasks will be kept in an ordinary room temperature. Now after a few days, we'll see that the seeds in the flask A will germinate. Clear? The seeds which is present in the flask A will germinate, but the seeds in the flask B, you can see over here, it does not germinate. So seeds in the flask A will germinate. So all the seeds which we are seeing over here in the conical flask A, they will show the process of germination. But the seeds which are present in the conical flask B, they will not germinate. Why? Because there is no oxygen. So what will be your observations or your conclusion inference? Your inference will that oxygen is necessary for germination. Okay, so your conclusion inference will be that oxygen is necessary for germination. Clear? So till now we have learned about the definition of germination. We have learned about the conditions for germinations, the things essential for germination, that is water, suitable temperature and oxygen, and even we have discussed the experiment, alright, to prove that water is essential for germination, suitable temperature essential for germination, and air or oxygen is necessary for germination. Now in the next part, we will see the next experiment, right, which is called the 3-bin seeds experiment. Is there in the page number 50? We'll go through it. Then we'll see the types of germination, the epithelial and hypothelial germination, and we will differentiate between the two types of germination. So in the part 2 of germinations, we'll begin with those topics. So this much for part 1 for germination. So we'll again begin with the part 2 of the topic germination under this chapter, seeds, structure and germination. Clear?